simple task turns into a time-consuming mess because work
Hello, Professor Dr. Samit Reddy. How are you? Please unmute your mic.
Okay, I was not allowed to unmute, but now I think we are good.
Yeah, it's working now. I make
you
you
hear
the
me?
co-host. Yeah, I can hear you.
Okay,
How are
thank you so
you?
much. And then I am the first one who is going
Yeah,
to present, right? Then I will share my presentation. Okay.
yeah. So we officially begin this session after 5 minutes. So you will officially open this session as a chair.
Okay. In 5 minutes, right?
Yeah, 5 minutes.
Okay.
3
Okay.
to 5
Okay.
minutes.
Okay. Sounds good.
What's time there?
It's 5 a.m. It's still dark. Yeah.
Thank you. We have successfully conducted our two first sessions. The first session was on poultry nutrition and alternative feed sources, and that was chaired by Professor Dr. Farina Khattak from Scotland's Royal College, UK. And there
Okay.
was four presentations from keynote speakers and two presentations from the graduate researchers. And
Okay. Very nice.
we were having the second scientific session on poultry diseases and gut health management. And that session was chaired by Professor Dr. Muhammad Kashif Salimi, Chairman Department of Pathology from the University of Agriculture, Faisalabad, Pakistan. And in that session, we will be joined by Professor Dr. Doc Corver, President Poultry Science Association, USA, and Dr. Anoop Kolanarjoni from the University of Minnesota, and Professor Dr. Don Kaspers, avian immunologist from the Monash University, Germany.
Very nice. Very nice. Yeah, I was not sure about the timing, if it is 4 a.m. or 5 a.m. because we have, I think, exactly 10 hours. You are 10 hours ahead of us. Yeah.
Yeah. Exactly.
We are good. We are good. How many attendees do you know? Do you have an idea how many?
We were having 25 plus participants in this meeting, and the conference is also live streaming on YouTube as well. And many of the people, they are watching on the YouTube as well. Those
very nice.
having not the meeting link.
Yeah. Thank you for inviting me. Thank you very much.
It's a great honor and a pleasure to invite you, such a distinguished personality in the field of molecular genetics and poultry science.
Thank you so much. Okay, it is 5:03 a.m. Should I go ahead or give 2 more minutes for people to settle down?
We are just waiting for one more keynote speaker.
Okay.
Then we will officially begin.
Okay.
Okay, so ladies and gentlemen, and thank you very much for joining this GRC 2026. We now move to our scientific session number 3. This session focuses on poultry genetics and genomics, and we have a very good lineup of guest speakers, in this session and 2 oral presentations from China as well. So, and this session is basically chaired by Professor Dr. Professor Dr. Samid Reddy from the University of Arkansas, USA, and he's also currently serving the PSA board as well. So, before we begin, let me give a brief introduction.
Okay, thank you so much, Dr. Akram and the organizing committee for inviting me. Good morning, everyone, or good afternoon from Arkansas, from the US. Yeah, it is a pleasure to be with you today, and I hope that we will have a wonderful session. This session focuses on emerging advances in protein genetics, functional genomics, epigenomics, host pathogen interaction, molecular breeding, and genome technology that are shaping, the poultry and the industry and the future of sustainable poultry production in the future. The session today brings, outstanding speakers. We have, I think, 4 keynote speakers, and then we will have 2 oral presentation. My understanding that the talk for the keynote speaker is 25 minutes, and roughly we will have 5 minutes for a question and answer. And those, I think Dr. Akram will handle them. I think my job today I will be the first keynote speaker to share with you some of the research that we conduct in Arkansas related to heat stress and, poultry, water conservation, growth, welfare, and so on. And then I will introduce, my the 3 keynote speaker that follows me, and then we will turn to the student. I think they are grad student, and I think the time 15 minutes for the grad student presentation, right?
Yeah, 10 to 15 minutes.
Okay. Yeah. With that being said, and without further ado, probably I should share with you my presentation. Sorry that I didn't send it earlier.
No issue. Yeah, you can share your slides now.
Okay.
So our first speaker of this session is Professor Dr. Samid Reddy. So just give me a brief introduction of Professor Samid Reddy. Professor Samid Reddy is an internationally recognized poultry scientist at University of Arkansas, specializing in poultry nutrition, heat stress physiology, feed and water efficiency, and metabolic disorders in broiler. His research focuses on understanding the molecular and cellular mechanisms underlying heat stress responses, woody breast, myopathy, and sustainable poultry production systems. In 2024, Professor Reddy received both the American Feed Industry Association Poultry Nutrition Research Award and the National Chicken Council Broiler Research Award at the annual meeting of the Poultry Science Association, recognizing the significant impact of his research on the poultry industry. His work on reducing woody breast severity has contributed to improving poultry welfare and minimizing economic losses in commercial production systems. He currently serves as the Poultry Science Association Board of Directors and is a scientific chair of the World Poultry Congress 2026. Professor Reddy continues to advance innovative research aimed at improving poultry sustainability, efficiency, and health worldwide. So thank you very much, Professor Dr. Samid Reddy. It's over to you.
Thank you, Dr. Akram. Thank you for the nice introduction. And again, hello, everyone. As I said, today I will share with you some of our research and scientific data related to heat stress. And today I will talk a little bit about something that most of the time we forget. I will talk about water efficiency today. But before to get there, I would like to remind myself and remind you about the importance of poultry production sustainability. Because in my mind, if I don't know the value of something, probably I will not appreciate the amplitude of the challenge that we are facing. Then I will share with you the importance of poultry production sustainability. I am sure everyone knows that. And then I will share with you briefly the challenges that we are facing, and then some of the potential solution. And those potential solution are based on data-driven and science-driven outcome. And then, we will open it for a few questions. If I cannot answer your question for whatever technical reason, you have my email address. Send me an email, and I will be more than happy to answer you. First, we know that the genetic selection for high growth rate and muscle development in the chicken has made spectacular progress. I give you an example here. In 1957, if we take this picture, nice picture I took from my friend and my colleague, Dr. Martin Swedoff from Canada. If we take 1-day-old chick in 1957 is about, approximately 35 grams. In 2005, it's 44 grams. You see, there's no big difference. However, after 2 months, that a chicken from 1957 would weigh almost 1 kilograms. In 2005, that chicken would weigh 4 kilograms. Then if you go from 44 grams to 4.2 grams, that's 100 times increase in body weight. Think about a little bit. If you translate that to a human being and you have a newborn child that weigh 3 kilograms at birth, after 2 months, that child would weigh 300 kilograms. It's a huge. This is show that efficiency of a broiler of chicken production. In the US, we produce yearly around 9.17 billion chicken. That production value is about $50 billion US dollars, and it provides more than 1.6 million a job. The chicken is highly consumed in the US, and also it's cheaper, is the less expensive, protein source. And as you see here, the projection for 2033, this production will keep increasing, and the consumption will increase also. And that's not only for the price, but also because the poultry is the most efficient food source. From 28, 35, or 42 days, you can have a chicken ready, or almost the cycle is short. It is relatively expensive, as I said. There is no religious taboo. I think everyone eat chickens. There's no issue there. It has a high organoleptic quality and high nutritive value, and we know that is lean meat. And also it uses low land requirement, and also there's a versatility in the production. Today we talk about organic chicken, we talk about pasture chicken, we talk free range chicken with conventional, and you name it. Then there's a high versatility there in terms of production. However, although this poultry production support the livelihood and food security of billions of people worldwide, it is facing several substantial challenges. The first one is based on the projected increase in global demand for food, which will be driven by the projected increase of a human population. Based on the FIU, by 2050, human population will be between 9 and 10 billion people, and that's required that the animal protein should increase around 73%. And from that, as you see here, the poultry probably needs to double the poultry production, and the meat and egg probably need to double that in 24 years. How we can do that? And we are facing today an increased risk of rapid transmission of animal disease. Today we talk about non-antibiotic ever, or raised without antibiotics. The increased incidence of metabolic disorder, as Dr. Akram mentioned, we have today muscle myopathy, from wooden breast to white striping to spaghetti meat to feathering tenders, and so on. Bone issue, lameness, and mainly induced by bacterial chondronecrosis with osteomyelitis. We have today also with the generation Z, we are oriented toward welfare and natural product, and we need to satisfy the consumer and their desire too. And also we have the adverse effect of extreme weather due to climate change and global warming. We have heat, we have cold stress, and also the increasing pressure on the availability of natural resources. Water, but I'll talk today about heat and water, but also food and energy and land. But today I will talk only about heat stress and water conservation or water efficiency. Just here to summarize, everyone knows the effect of heat stress. The heat stress is well known, welfare, economic health, and the production burden. In the US, this paper is from 2003, showed that in the US, every summer or every year, due to heat stress, we lose between $165 to $185 million. And I am quite sure the number today is much, much, much higher. As I mentioned, we've seen large, abrupt, and widespread heat waves that have occurred in the past. I give you an example. In France, 3 years ago, 5 million chicken and turkey died in 1 week when the temperature reached 40 degrees Celsius. I am quite sure that you remember in India in 2015, due to heat waves, 17 million chicken also died, and so on. Based on the Environmental Protection Agency, they estimated, this is published in 2013, that by 2100, the average US and the global temperature are projected to increase between 2 and 5 degrees Celsius. Then we will face extreme heat waves, widespread, and more frequently. And one thing also is associated with the climate change and global warming and drought condition is the water scarcity. I've been in Peru and Mexico, India, North Africa, and the water just for human drinking and human consumption is an issue, and you see it everywhere in most of the high-ranked, scientific journals, Science, Nature, and PNAS, in the news, BBC, and so on. Then the water scarcity is an issue. It is a looming international issue. As I said, the global climate change in tandem with the land use change will raise problem. It's already raising a problem for water and food availability, not in terms of quantity only, but also in terms of quality. Today, we have 2/3 of the global population, they live under condition of severe water scarcity at least 1 month a year. And based on the FIU and the Stockholm Environment Institute, they estimated that by 2080, 6 billion people will live under severe water scarcity. In the US, most of the time we take things as granted. Yesterday we have a really nice, rain here in the US, but this winter we have only 1 day snow. Then based on the US Government Accountability Office, the GAO, like we have 40 out of 50 states will have at least one region that they will have water scarcity in the next decade. Then from my opinion, and this is me, it is time to rethink and to reevaluate our approach to water. If you see in the NRC table of nutrient, water is the most important nutrient, but you never see it in the table. 80% of our body is water. Water is the most digested, water is the most consumed nutrient, but it is forgotten. We never measured. I don't know, probably because we don't have the tools to measure water, with accuracy. But it is the most important nutrient, but it is overlooked or forgotten. Today, I would like to share with you is the cooling water. The water usage to keep the birds cool during summertime. We have today, we use cool cells, and although the seminal managerial, nutritional, and genetics work that has been conducted, we still have, we still lose, we have a high or significant economic loss due to heat stress because the production efficiency declines. In my mind, I think there are two reasons. First, the molecular and cellular mechanism that are involved in heat stress response is not well defined. We're still working on that. And also some of those approaches, whether management or nutrition, they have been applied without fundamental knowledge. And I hope I will show you a few examples. Okay. The status quo pertaining to keep the birds cool during hot weather, whether evaporative pad, we call it fan and pads, or the misting system or the fogging system. The first one, it uses a large amount of water. Based on the National Poultry Technology Center in 2015, they estimated that in the US, that water usage using the cool cells can reach 11,000 gallons per broiler house per day. 1 gallon is 3.7 liters. If we have 75,000 broiler houses in the US, and we have more, that water usage per day can reach 825 million gallons a day. That's a huge amount of water use. The second system, the fogging system, which is meant to keep the barn cool, that system for every 1 degree Fahrenheit of cooling, when it reduces 1 degree Fahrenheit, it increased the relative humidity by 2.5%. Now, if you combine heat stress and high relative humidity, that is a catastrophic situation for the chicken. The chicken cannot handle that. Okay, I have two colleagues, Dr. Liang here from University of Arkansas, and Dr. Tabler. He's now in the Tennessee University. In between 2009 and 2011, they conduct research using 5 flogs at a commercial level, and they used what we call the sprinkler. The sprinkler is not the fogger, but instead to cool the barn, it spray big or large, I don't know the dimension, large droplet of water intermittently every 5 minutes or 10 minutes based on the body weight and the temperature of the barn and so on the chicken, which means this system is meant to cool the bird rather than the barn. And what they see, there's no difference in term of mortality, as you see here. However, the sprinkler, I call the sprinkler here, and they compared with the cool cells, the conventional one. The sprinkler improve the FCR by 6 points, as you see here in this table on the right side. No effect on liter moisture content. However, the most important thing that this sprinkler saves 66% of water usage. 66% of 825 million gallons a day. That's a lot of water conservation. It's beautiful. Then they approached me. I said, okay, it has an effect, but today we need to go for mechanistic understanding. Yeah, you have an effect. And as I said, what is the mode of action? Then we repeated at the experimental level. Here we have a nice environmental chamber in Arkansas. We have 12 environmental chamber, 24 pans, and we can control the relative humidity and the temperature. It is well controlled. And here we did 3 treatment. We have thermometer condition 24 degrees Celsius. We have heat stress, and then we have heat stress with the sprinkler. I didn't compare the cool cells here. As you see here in a black line here, that's the thermometer condition. The core body temperature is around 42 degrees Celsius or 41.5. We use eye bottom to measure the core body temperature. And the red one, that's heat stress. This is as expected, increase the core body temperature. And in the blue here, it's the sprinkler. We see that the sprinkler reduce significantly the core body temperature induced by heat stress in chicken. And the same for the surface temperature here. We used infrared camera here. And as you see, this is heat stress. This is heat stress plus sprinkler. You see it also alleviate that core body temperature and surface temperature. In terms of growth, as you see here in red, this is heat stress. We know heat stress depressed feed intake. The blue, the thermometer condition. The green is the sprinkler. Then the sprinkler alleviate that core body temperature, that stress, and then improve feed intake, improve body weight gain, and also modulate the time we call the feeding behavior, the time spent eating, and the frequency to the feeder. One more thing, as you see here, heat stress, we know that heat stress increase water consumption. The sprinkler reduced that water consumption. That again, the sprinkler seems not only to save 66% of water use, but also reduce the water consumption because it reduced that core body temperature and that stress. We try to understand, we know that the thermoregulation and the appetite and water consumption are all controlled by the brain. First, we measured the circulating level of corticosterone, the gold standard for stress. As expected, heat stress increased plasma corticosterone level. The sprinkler reduced. This is again, the sprinkler seems to alleviate the systemic stress by reducing the corticosterone level. And then we measured the heat shock protein, as their name mentioned, heat shock. They are chaperoning and they are induced by heat shock or by any type of stress. As you see here, heat stress in the brain, the hypothalamus, increase the expression of heat shock protein 60, 70, and 90. The sprinkler reduce that expression. From here, we can say that the sprinkler reduce the core body temperature, reduce the surface temperature, reduce the systemic level of corticosterone, and also downregulate the expression of heat shock protein, which means also downregulate or reduce the stress in the internal organ and the hypothalamus, as an example here. Okay, we know that feed intake also, it is controlled by the brain, but at the level of the brain, there's several nuclei involved in feed intake regulation. There are several neuropeptides. We classify in two categories. One is orexigenic, like stimulate appetite, that is under the control of neuropeptide Y and agouti-related peptide here in green. The other one is anorexigenic, is inhibit feed intake and appetite. It is under the control of POMC, pro-opiomelanocortin, and CART is the cocaine amphetamine regulated transcript. This is in normal condition. When you are hungry, the NPY is upregulated and agouti-related peptide. Then you eat. When you finish, then the POMC is upregulated, and then you stop eating. This is under the normal condition. As you see here, heat stress upregulate the hypothalamic expression of POMC, and that's expected. Then depressed feed intake. And when we use the sprinkler, the POMC expression is downregulated and in heat stressed abroad. That's explained probably that the regulation of appetite and growth. The second marker that we measured is the orexin. Orexin, as its name mentioned, orexin is orexigenic, stimulate appetite in mammalian. In chicken, we don't know yet the effect of orexin, because when they inject orexin by ICV, there's no effect on appetite. But my guess that orexin is also involved in stress. My guess that, you know, when orexin is up, it induces stress, or it is induced by stress. And here, as you see, when we have the sprinkler, the orexin expression is downregulated. From here, we can say that the sprinkler modulates that appetite, feed intake, and the growth via the modulation of POMC, neuropeptide POMC, and probably stress via orexin. The last marker that we measured is the AMPK, is the adenosine monophosphate protein kinase. This is the energy sensor. This enzyme is controlled or is regulated by the ratio of AMP and ATP. If you have enough energy in your cells, this enzyme is not affected. However, when the level of AMP is up and your energy level ATP is down, that ratio increase. This enzyme is activated by phosphorylation at the threonine 172, and when it is activated, it will shift the anabolic pathway to catabolic pathway. It will block any synthesis and start using, you know, the catabolic pathway to provide energy to the needed for the cells. As you see here, heat stress activate AMPK. The sprinkler bring it down. This indicate first that, you know, stress. Because heat stress, when the chicken are stressed, they divert the blood to the skin, right, to eliminate that excess of heat. But when the blood is diverted to the skin, you have less blood that goes to the internal organ. And if you have less blood go to the internal organ, which means you have less oxygen, probably is a hypoxic condition, but you have also less nutrient. In addition, the chicken eat less. Then you have less blood and you have less food. It makes sense that you have less energy. And when we have less energy, then that AMPK is activated. That's explained here, the upregulation of AMPK in heat stress. This sprinkler bring it down. And this indicates also that sprinkler alleviate stress, but also preserve the intracellular energy that the cells need. To sum up this one, then we see that the sprinkler saves 66% of water, reduce the core body temperature induced by heat stress, the surface temperature, alleviate the systemic and intracellular stress, and preserve energy, and use or promote growth and body weight, which mean probably also promote also the enhance the welfare of the bird. We have two paper here. If you have time, you can read it. The next one, it is the drinking water. I talk about water usage, now drinking water. Here in collaboration with Dr. Orlowski and Dr. Nick Anthony, my colleagues. Dr. Nick Anthony now is with Cobb Genetics. We selected two chicken line divergent selection based on water efficiency. And here we use the same analogy as feed efficiency. We used feed conversion ratio for feed efficiency. Here we used water conversion ratio, how much they gain based on what they drink. Today we have generation number 9. Here we expose those chicken. We used 2x2 factorial design. We expose them to thermonuclear condition or heat stress. And the first thing we measure is the core body temperature. But one thing is striking here and surprising, as you see here, from day 28 to day 30, in red, that's the high water efficient bird, which means the high water efficient, they have lower water conversion ratio, which means they drink less water. The high water efficient line, they have high core body temperature. And that difference, that amplitude from day 46 to day 48 become bigger and bigger between the two lines, which means under heat stress condition, the high water efficient birds, they have high core body temperature. However, even with that, they consume less water. That's why they are selected for high water efficiency, as you see here in red. Here in red, under heat stress, the high water efficient, they drink less water, even compared to low water efficient bird under thermonuclear condition. They are so efficient, and they eat less food, but they have better FCR. As you see here, the FCR under thermonuclear condition, they perform better, 6 points better, and under heat stress condition, they have 9 better FCR point. Or the water conversion ratio, they have 59 better WCR and thermonuclear condition, and 66 point under heat stress. Then they become more efficient, not only in terms of water consumption, but also in terms of feed efficiency. And here, what I am going to challenge you. We used to say, okay, for every 1 gram of feed, the chicken drink probably 2 grams of water. I think today that the water consumption override the feed consumption. Because if the chicken does not drink water for several days, it will die. But they can survive without food. I think today, it is the opposite. The water, if you are efficient in water consumption, you become efficient in everything, including feed consumption. Then we did several molecular analysis at the level of the brain to understand the molecular mechanism that underlies that water efficiency. We found several systems, the AVP, the arginine vasopressin system. We also found some differential expression in terms of the RAS, the renin and angiotensin aldosterone system, and also the aquaporin system. We also found that the high water efficient bird, they have low level of FITC in the serum. The FITC, we use the FITC to assess the gut integrity. They have lower level of serum FITC, which indicates that this bird, they have better gut integrity too. With that, we have several papers in terms of here, the one in physiological reproduction about the molecular mechanism at the level of the brain that regulate water homeostasis. And we have a 3 paper in frontiers and poultry science about the gut integrity. We use the different segment of the gut. We use the duodenum, the jejunum, and the ileum. And I invite you to check this paper. It is 5:34 a.m. I think my time is due. I hope in the future I will talk about the nutritional component management. We used microalgae, and we alleviate stress. I would like to thank my team. I would like to thank the funding support, my collaborator, and thank you all for your attention. And I will be more than happy to take any question that you may have.
Thank you very much, Professor Dr. Sami Zeri, for such a wonderful presentation. So right now,
Should I stop sharing? Should I stop sharing?
Yeah.
Okay. Good.
So now, if anybody have a question from the audience, you can unmute yourself or raise your hand, please. Ali Hassan Nawaz, do you have any question?
Yeah, I have question from Professor. First of all, I want to thank Professor Sami for her beautiful, very knowledgeful presentation, because I'm also working on heat stress in Nanjing Agriculture University, and I am following his work. So his work helped me a lot during my research. So Professor, I have a question about you worked on the NPY, like the neuropeptide in the hypothalamus, that how it relates to the heat stress. So, but can we study something like we can have some GWAS kind of analysis, or like selection sweep? We can compare in the red jungle fowl the genetics behind this mechanism, the neuropeptide.
I've heard neuropeptide, but you wanted to compare the neuropeptide with what? I didn't hear the question very well. Can you repeat, please, the question?
Yeah, Professor, I want to say because we know we studied the physiology of this system, but we don't exactly know the genetics behind this. So there are many indigenous breeds. So can we conduct some study kind of GWAS or selective sweep analysis to find the exact?
Yes, absolutely. There's the 2 things here.
Yes.
The system that I showed that they are involved in water homeostasis, the aquaporine, the RAS system, and the AVP. We compared the expression of those systems in 4 population of chicken: jungle fowl, which is the wild type, chicken selected in 1950, we called ACRB, chicken selected in 1995 and 2015, 20 years between them. And we see some difference. But we didn't compare the neuropeptide. We didn't compare the expression of NPY, POMC, orexin, and all those. You can compare, but keep in mind, those neuropeptides, they are also expressed in other tissue too. They can be expressed in peripheral tissue too, like the gut, for example.
Okay. Thank you, Professor. Another question I have, because I am also, because the concept of this water scarcity is very interesting. So if because my work is majorly related to the selective sweep analysis and then RNA-seq and eQTL mapping. So I'm doing this kind of work. So how can we relate water scarcity with the FCR, and how can we find some genetic regions or genes that might be?
I hope to answer your question. I hope next year I will have the answer for your question. We are working now on GWAS and QTL. I hope we will find some chromosomic region that involved in that water efficiency. I hope.
So this work can be done, like we can sequence some indigenous breeds, and we can see like which breeds are more water efficient, because we already know the efficient, but we don't know the water efficient chicken breeds.
Yeah, my suggestion, if you have those breeds and you have the phenotypic data, if you have the water consumption, accurate water consumption in different condition, yeah, you can take, you can take some whether from blood or from tissue, DNA, and do some GWAS, and you will find something, yeah. SNP and
Okay,
yeah.
okay, Professor. Thank you so much.
It's my pleasure.
It's really nice.
Yeah. Dr. Akram, you take it or I go?
Okay, thank you very much, Professor Dr. Sami Dredi. So before proceeding further, I would like to inform all the participants that Professor Jozo Song, Professor of Avian Genetics and Genomics at the University of Maryland, USA, just sent me an email. He was unable to join us today due to unforeseen internet connectivity issues while he is traveling in China. So we may proceed with the next presentation with Dr. Kate Sutton from the University of Edinburgh, UK. So, Professor Dr. Kate Sutton, are you on the board? Can you hear us?
Hello, can you hear me now?
Yeah,
Yes.
perfect.
Yes, perfect. Hi, everybody. I share my slides. Yes.
Dr. Akram, would you like to introduce the speaker?
Yeah, I would like to introduce her,
Okay.
giving her a very brief introduction. Okay, so before Dr. Kate Sutton begins her presentation, let me give a brief introduction. Dr. Kate Sutton is a career track fellow in avian immunology at the Royal Dick School of Veterinary Studies, the University of Edinburgh, United Kingdom, with close affiliation to the Roslin Institute, UK. Her research focuses on the biology and functional diversity of innate immune cells in poultry, particularly macrophages and dendritic cells, and their role in natural and vaccine-mediated immunity. She investigates immune cell heterogeneity in chickens and how these populations contribute to protective immune responses against infectious diseases. Her work integrates advanced immunological and molecular tools, including transgenic chicken models, multicolor flow cytometry, unfocal microscopy, and transcriptomic approaches such as RNA sequencing and single-cell analysis. A key objective of our program is to identify immune correlates of protection that can inform improved poultry vaccine design. In parallel, she is internationally recognized for developing and applying chicken organoid systems, which serve as physiological relevant in vitro models of avian intestinal and respiratory issues. So with this, I would like to ask Dr. Kate Sutton to please begin with your presentation. And thank you very much for joining us.
Lovely. Thank you very much for the introduction. And hello from sunny Edinburgh, which is quite rare. It's usually raining. And so today, yes, I'm going to talk about the work we've done at the Roslin Institute on the development of 3D organoids. And before I begin, I'm just going to give a little bit of background about organoids and what we know about farm animals and what we know about the chicken organoids. So to begin with, you can have two types of organoids. You can have an organoid that's developed from a pluripotent stem cell. So here you can take these cells, give them some growth factors that will lead to them developing these 3D structures. Or what you could do is get an enteroid. You sometimes see enteroid used in the literature. It's interchangeable, enteroid and organoid. But essentially, the difference is that these are from the actual tissue, an adult stem cell, or you can get like quite young stem cells. And what you do with both types of cells is that you put them into this matrigel, which is an extracellular matrix. And this is comes from like tumors formed in mice. So it is an animal-derived product. And when they're put into this matrigel, what you get is these kind of 3D structures that start to resemble the tissue from their origin. So you can derive them from the intestine, you can have them from the liver, you can have them from the lung, the brain, the eye. So there's many different organoids that you can have from different tissues, but we're going to concentrate on intestinal organoids because that's what we have for the chicken. So what happens is over time, when you seed these stem cells, they start to grow into these 3D structures. If you take them from the intestine, they're going to consist of the epithelial cell lineages that you find within the tissue itself. So that'll be enterocytes, goblet cells, Paneth cells, stem cells, and so forth. What they'll also do is form these tight junctions between the epithelial cells. So epithelial cells form that barrier, that physical barrier from the environment into your intestine. And what you want to do is with these epithelial cells, they should form this kind of wall. And these are produced, this kind of wall formation is made by these tight junctions between the epithelial cells. So these kind of 3D organoids from the intestines really resemble what you see within the actual species in which they're derived. When it comes to farm animals, using the same protocol that you use for mouse and humans, so you take your scripts from the intestine, you put them in the matrigel, you put all these different growth factors depending on the species, and within a couple of days you get these 3D we call it 3D multi-lobulated structures. Essentially, they're nice, big, they've got these little arms coming out of them, and what you can do over time is you can start to passage these. So what you do is you break them down by just simply mechanical destruction, and they'll form individual crypts again, and then go on to proliferate into these 3D structures. So these can be kept passaging for up to a year. So they're quite good models to have. Now, when you use the same protocol in chicken, you take the crypts, put them in the matrigel, put all these fancy growth factors, you can get these 3D structures. So this is one of the earlier studies in 2012 showing these organoids in the chicken. Now, they weren't great. You can see from the previous images that these don't have these kind of like arms coming out of them. But a more recent study was able to successfully apply the system that they use in mammals to the chicken, where you can see that they form these kind of 3D structures. But what you do find is that they don't look as big as what we see in other species. So in the previous pictures, you can see that they're quite big and quite multi-lobulated. So it seems that for the chicken enteroids, or essentially the intestinal organoids, they don't really grow in the kind of the way that you can with a mammal one. However, at Rosalind in 2021, we had a PhD student called Tessa Nash, and she came along and said she was going to develop the 3D organoids in the chicken. And what she found was she could take the intestine from embryonic 18 and 19 embryos, chop them up essentially, and she released these kind of villi pieces. And what she found within 24 hours is that these pieces started to close up into these 3D structures. So I'm going to talk a little bit about polarity here so that you can understand the kind of how different the chicken organoids are. So when you take an organoid from a mouse and you grow it in the matrigel, what happens is your epithelial cells grow inwards, essentially. So they all face inwards, and this would be considered, the core of the organoid would be considered the lumen of the intestine. So this is what we call a basal out organoid. So when you stain them with F-actin, you see here the F-actin stains the kind of surface of the epithelial cells. So they finger-like projections that are on the top of the epithelial cells. So what this means is if you were to come along and say, well, I want to look at host pathogen interactions, or I have a compound or a feed additive I want to look at, and I want to apply it to the organoid like it would go through the stomach and into the intestine of an animal, you would have to get your compound into the core of the organoid here. And there are many ways you can do that. One, you could do a microinjection. So you can come along with a very fine needle and inject your compound of interest into the core of the organoid. Now, the only problem with that is quite cumbersome. It takes a lot of training, but also you can't really get the same dose across each of the organoids because they differ in size. So some can be quite small, some can be quite big. So it's quite difficult to get your dose right. The other thing you could do is mechanically break up your organoids. So here you can break them into little chunks, just a triple E or mechanical breakage, and you release the kind of cells and add your compound of interest, and then these will form back into your organoid over time. The only problem is then you're exposing both the apical and the basolateral region of your epithelial cells, which is, if you're looking at something that's coming in through the oral route, that's not what's going to happen. They're going to actually interact with the surface of the epithelial cells to cross over into the tissue. Another thing you can do is actually digest your organoids into single cells and then place them onto a surface of matrigel, and they'll form a 2D layer. And this is really good, but it can be quite cumbersome as well. I've had experience doing this in cattle, and it's not really consistent, and you can't keep the cells growing for very long. They can last up to a week, but it means you also have to continually keep the 3D organoids growing and grow up lots of them to make these 2D cultures. Now, with the chicken organoids, or actually, before I begin, With the mammalian organoids, there's another thing you can do, is you can actually change the polarity. So as I said, you can have these basal out organoids. You can take them out of the matrigel and put them into this kind of special, not really special, but a media containing EDTA. And what they found is that you have these basal out ones can turn into apical out organoids. So you see here the F-actin is enriched within the core, but when you take them out of the matrigel and grow them for a couple of days, they flip outwards. So now we have an apical out organoids. And what this means then is that you simply can just add your compound of interest to your cell supernatant and just kind of not have to worry about dosage or worry about microinjection or worry about contacting parts of the epithelial cells that you don't actually want to target. There are disadvantages to this, is that if you have 100 organoids, 90% of them will flip inside out. Another 10% will actually not reverse their polarity. And I think they can last up to 12 days in culture, so they're quite limited. So you still need to keep your basal out organoids growing. For the chicken organoids, what Tessa found is that if you take the villi and you grow them without the matrigel, so essentially they're just put into a cell culture media, they do not reverse their polarity. So they stay as an apical out organoid. Okay, so here is an image here of a chicken organoid with the F-actin, and you can see the enrichment of F-actin binding to the outside of the organoid, meaning all your epithelial cells are facing outwards. So this makes it really easy then to come along and add your compound or your pathogen of interest straight into the cell culture media as if it was a 2D culture. So the 3D organoids don't require the matrigel, which is quite expensive and is an animal-derived product. They're facing outwards, which makes working with them quite handy. And yes, essentially that's my advantages. But the other thing we notice about them is that you'll see that they have these inner core of cells. So there's quite an abundance of cores, cells within the core of the organoid. And what Tessa found is when she stained the organoids for cell markers for immune cells, such as T cells, B cells, macrophages, she found that the inner core of the chicken organoids contains these cell types. So recently we did a single cell RNA-seq study on the chicken organoids and found that they're full of many different cell types. So you've got the epithelial cell types, you've got the mesenchymal cells, which is smooth muscle cells, fibroblasts, and so forth. But really interesting, they also contain immune cells. Because they're derived from embryonic day 18 intestines, they consist of the cells that we expect to find within the intestine at that point of development. So it has macrophages, heterophils, NK-like cells, and gamma delta T cells. So really, the chicken 3D enteroids resemble the actual architecture and the cellular heterogeneity that you find within the tissue itself. So really, really, really good and unique model. The one thing you can do with the chicken 3D enteroids is also make them into 2D organoids. So you can just digest them simply with EE Express, and you get in and seed them onto a matrigel coated plate, and you can get these 2D structures. So the one thing when Tessa first developed this model, and then we had the 2D model developed, we were saying to ourselves, well, how can we use two of the models to look at the interaction of immune cells and epithelial cells within the models? So what we did was we took the 3D organoids and the 2D organoids and we infected them with Salmonella typhimurium. And in this study, we used a Salmonella typhimurium that had a GFP tag on it, making it easy to visualize in the models. So in this top model here, you can see these are the 2D organoids, where you see the Salmonella being infected, and you can see they're quite easily infected with the bacterium, and also with the 3D organoids. Again, you can see they become infected. Our next question was, the 2D enteroids don't contain immune cells, but obviously the 3D enteroids do. So what we were able to do was, can we distinguish the contributions of the immune cells to an infection, and also look at what the 2D only, epithelial cell only cell culture model is contributing to the immune response? So in a very quick answer, or quick kind of look, this is an array of innate immune-related genes that we have. We have a little chip at Roslin called the Fluidigm, allows us to measure 96 genes across 96 samples. And here I'm showing the genes that are significantly differentially expressed and shared between the time points and shared between the models. So you'll see here at 4 hours post-infection, you have a lot of genes that are upregulated in the 2D model. If you remember, these guys don't have immune cells present, they're just purely epithelial cells. Whereas in the 3D model, where we see both epithelial cells and immune cells present, we do see an upregulation, but the upregulation is much lower in comparison to the 2D. However, by 8 hours post-infection, the immune response is dampening. It's decreasing in the 2D organoids, but increasing in the 3D organoids. So we've shown here that the two models are really great for looking at the contribution of immune cells, but also the crosstalk between immune cells and epithelial cells in the model, and for looking at host pathogen interactions. In the next example I'm going to show you is the use of the organoids in study of avian influenza. So we all know that avian influenza H5 and H7 high path are notifiable diseases. So once there's an outbreak, they're known to be H5, H7 and known to be high path, they had to be notifiable to the authorities to essentially get parameters in place to reduce the outbreak of disease more. However, there has been incidents of low pathogenic avian influenza strains causing mild to moderate symptoms. So the moderately virulent low paths can cause up to 5 to 95% mortality in young chicks and egg-laying hens. And these have been found in the strains of H3, H6, and H9. Unfortunately, when these outbreaks happen, they're non-notifiable because they're not high path, they're not the H7, H5 strains. So what we want to do was look at ways of if there's an outbreak of low pathogenic avian influenza with unknown virulence, can we use the 3D organoids to see whether they're going to cause like kind of moderate to low disease, or are they going to end up being strains that can cause harsh disease or what would you say, high disease in the animals? So what we did is we took 3D organoids from broilers and layers, essentially. So we took two different genotypes. And what we wanted to look at was if we infect them with different types of avian influenza viruses with known virulences, can we look at differences in the organoid model at that level? So I should say that I go through the viruses before I begin, but the H3N1, this is a strain that was an outbreak in Belgium that caused high mortality in egg-laying hens, but it did cause disease in broilers, but it was moderate disease. For the hens, it's tended to cause them to reduce egg laying and become quite ill. For the H5N2, H6N1, these would be mildly virulent strains. And then we have a H9N2, which is an avirulent strain. So what we did was take each of these strains and infected enteroids derived from a broiler or a layer line organoid and looked at the outcomes of that infection. So the things we looked at was the location of the virus. Was the virus able to infect the organoids? Was it able to actually travel cross the epithelial cells to the inner core of the organoids, which represents the lamina propria of the intestine? Was there a difference in immune responses in the organoids? So essentially, if you have an organoid from a layer or from a broiler, what's their responses to these pathogens or to these viruses? And can we look at genotype to phenotypical differences that we see within the field? So to begin with, I'm just going to show you some images that we have. So this is your mock control here. This is an unaffected organoid. And then this is a H3N1 infected organoid. This is the virulent low-path avian influenza virus. And you can see here in green is the virus. You can see it's able to infect the epithelial cells quite happily. So this is the surface of the organoid. So here I should say this is the inner core of the organoid. So here you're looking at in the middle of the organ. Because it's a 3D structure, you've got to go through them to take an image. So this would be the lamina propria, and these are your epithelial cells. This is the top of an organoid, so essentially looking down on top of it, and you can clearly see that the surface of the organoid is highly infected by avian influenza. And this is then just kind of a zoom in of that image where you can clearly see the epithelial cells are infected with the virus. So this is for the H3N1. And you can see here when we go into the core of the organoid, there's not much virus present. So it's able to infect the epithelial cells, but not able to actually get into the lamina propria, at least at 24 hours post-infection. For the other virus that we have, if you remember, the H5 and the H6 are mildly virulent strains. These two are able to infect the organoids, but again, we only see them in the epithelial cells on the outside and not in the lamina propria. And for the H9N2, if you remember, this is the avirulent strain, and this also had the ability to infect the organoids, but again, at the surface and not in the lamina propria. So the next question we had was, if we looking at the replication of the virus, this is a qPCR, looking at the replication of the viruses, is there a difference in the virus's ability to replicate, whether it's in an organoid derived from a layer or the organoid derived from a broiler? And we can clearly see for the H3, H5, and H6, there's no difference. They can replicate at the same level. The only statistically significant difference we've seen was, surprisingly, with the avirulent H9N2. It seemed to replicate at much higher levels in the broiler-derived organoids in comparison to the layer-derived organoids. So the next I want to look at is what is the difference in the immunological responses of the organoids to these different viruses? So can we see whether the viruses are inducing particular responses in the organoids like what they do in vivo? So as I said, this is a measurement of 96 innate immune-related genes on our Fluidigm chip. So what you see here is these pie charts, and in red is the number of genes that are significantly differentially regulated out of the total number of genes. So in blue are the genes that they were regulated but not to a statistically significant level. So for the H3N1, you can see with the broiler-derived or the layer-derived organoids, there's not much difference. There's 8 genes that are differentially regulated in the broilers, more in the broilers than in the layers. But surprisingly, with the H5N2, you see that the layer-derived organoids are not actually responding quite well. This is at 24 hours post-infection. So you can see only 5 genes are differentially regulated, but a huge amount more genes are regulated in the broiler-derived organoids. We see the same thing trend in the H6N1 infected organoids, that there seems to be the broilers that have a bigger response, and with the H9N2. So what does this mean? So our next question was, well, let's look at the genes that are regulated within the actual genotype. So here we have a Venn diagram that's looking at the genes that are regulated across all viruses, essentially. So here are the genes that are upregulated, if that makes sense. So for the H9N2, these 4 genes are unique to that organoid when it's infected in the layers. For example, only one is unique to the H5N2. Only one gene is shared across all of the viruses. So when you infect the layer organoids with these 4 viruses and look at the genes that are individually, and look at the genes, interferon lambda, which is a gene that is quite linked to epithelial cell immune responses, is the only one that's shared and regulated, but it's upregulated in the models. When you look at the cells that are, I'm missing the word here, but downregulated, you can see that in the layers, this H3N1, this is a virulent low path, it is actually inducing a downregulation of the immune response in the layers. And if you remember, I told you that the H3N1, this affects the layers more than the broilers, okay? So when we go into the broilers, we're seeing the opposite effect. So instead of downregulating the immune response in the broiler-derived organoids, this H3N1 is actually upregulating a lot of the genes, and not many genes are being downregulated. So even in the organoid models, we can see a genotype to phenotype variation in their responses to different low-path avian influenza viruses of different virulences. So overall, I hope I've shown you that both the 2D and the 3D models can help discriminate the contributions of the different intestinal compartments to host immunity. As I said, you can distinguish the epithelial responses and epithelial cell contribution to the immune response, but also can look at the epithelial and immune cell crosstalk in the 3D organoids. We've shown here that the chicken organoid models have the capabilities to investigate genotype to phenotype variations, especially in response to avian influenza viral infection. And they could possibly be a model that could be rapidly implemented during the emergence of novel avian influenza viruses. Because at the moment, if a virus is a H5, H7 high path, you go into an actual animal. So it's quite costly and quite labor intensive. So maybe the chicken organoids could be used to kind of rapidly test whether an unknown virus is what its virulence could possibly be. And they can be used for many different things to determine host factors, enzymes that improve virulence or decrease it, and so forth. So I'd like to thank everybody in my group for help with this work. It was a flying visit on organoids. To Paul Dickard and Wayne Min, who provided us the avian influenza viruses, and Shakti Witt at GD, who also provided the viruses for the work, and Lonica, who obviously developed the model, has now since moved to GD, and past members of the group, and to the NAR for all their help with animal studies. Thank you very much. Any questions? Sorry.
Thank you very much, Dr. Kate Sutton, for such a wonderful presentation. Professor Sami Trivedi, it's over to you, please.
Yeah, thank you, Dr. Sultan. It's a really nice presentation. I will start with one question.
Yeah.
What are the conditions that temperature is 37 or 38 degrees to culture those organoids?
We do them at 37. Because these are embryo derived, we think they can't cope with high temperatures. So we have tested the 41, 42 degrees and they just disintegrate. So that's not to say that if you derive them from adult birds, they might work better in higher temperatures. So we do have a study ongoing now trying to look at using the models for environmental stress. So using heat as a stress on the organoids and looking at how that affects susceptibility to disease and how that affects metabolic related genes. And we're now seeing that they can go to 38, 39, not so much. So yeah, you're quite limited in your temp, but yeah, they are 37. We found works the best for these guys.
I will be in touch.
Good. Hope so.
I am working with heat stress, and I think you have a wonderful model to do that.
Yeah. No, it is. Yeah. Happy to share what we've done and help in any way. Yeah.
Yeah, I'd love to collaborate with you in the future, and I will be in touch. Is that okay with you?
Good.
Okay. Any other question for Dr. Sutton?
Any other question from the participants? Okay, Professor Sami Dridi, should we begin with our next talk?
Yes, please. Yeah, let's move to. Thank you, Dr. Sutton. It was really
You're welcome.
nice.
Thank you very much. Yes, thank you.
Thank you very much, Dr. Kate Sutton. We have a certificate of appreciation at the end of this session for you. Please, if your schedule allows you, stay with us at the end of this session. Thank you.
No bother.
Thank you. Okay.
So next talk we have Dr. Walt. Nice. Walt. Yeah, Dr. Akram, you take. Yeah, go ahead.
Okay. So our next keynote speaker is Dr. Anna Wall from Iowa State University, USA. She will present on breeding chickens for sustainable egg production. So Dr. Anna, can you hear us? Are you on the board?
Hello? Is it working?
Yeah.
And can you see my screen?
Working. Yes, we can see your screen too.
All right.
Okay, so let me give a very brief introduction to Dr. Anna. Dr. Anna Wallach is an affiliate assistant professor in the Department of Animal Science at Iowa State University and a leading genomics geneticist at Highland International. She is highly recognized for bridging academic research and industry applications, specifically utilizing advanced genomic technologies to transform commercial poultry breeding. Dr. Wallach has co-authored roughly 190 scientific publications, receiving substantial citations within her field. Her work has earned prestigious awards from the Poultry Science Association, notably winning the American Egg Board Research Award twice. So thank you very much, Dr. Anna, for joining us today. It's over to you now.
All right. Thank you very much for your kind introduction, and thank you everyone for being here. So Dr. Didri started with the broilers. I'm going to move us to the layer side and talk about why to work on sustainable egg production. So I will have quickly go over 6 different parts. So get some context and then describe different compartments and parts of the my definition of what sustainability in layer chicken means, and we'll finish with a little bit of summary. So if you talk to any researchers, you probably find as many definitions of sustainability as there are researchers. So what I'm going to use today as the definition of sustainability is simultaneous improvement in 3 different aspects. One of them is animal welfare, and then environmental efficiency and economic resilience. And we have to work on all of these 3 aspects together at the same time. So why is it important for layers? Dr. Didri said how much impact the broilers have. The layers produce about 90 million metric tons of eggs, and there are approximately 1.5 billion laying hens in the world. So that is a lot of chickens. Even though they are small, their sustainability still can have a large impact, because for every kilogram of egg protein, they are going to excrete or use up about 8 kilograms of CO2. But eggs are really valuable food for humans. They deliver complete protein with all essential amino acids and key nutrients. And relative to the other livestock species, we are already doing really good in the sustainability, because eggs produced about 10 times less greenhouse gases than the beef, and require also about 10 times less land. But because the eggs are such a valuable food for humans, we expect global egg demand to grow by about 30-35% in the next few decades. So the production in a sustainable way is getting even more important. So a little bit more about those 3 pillars of the sustainable layer genetics. So the first group of traits will be related to animal welfare, and we have here livability, because if you're a dead chicken, it's kind of hard to even talk about your welfare. And then we will talk about injurious behaviors, kill bone integrity, fear, and physiological stress resilience. Then from the environmental side, the main trait is some kind of feed efficiency, which can be expressed as feed conversion ratio or residual feed intake, and also persistency of lay and nutrient excretion per egg. From the economic side, of course, the layer chicken has to lay lots of eggs, but then also a critical trait is shell strength, because this will allow the eggs to actually get intact to the customer. So we'll talk about egg quality and robustness to different environments, resilience and resistance to diseases and heat stress, and also ability to produce on different diets. So similar to broilers, where genetics had huge impact in the last few decades, we can see similar large impact of genetic improvement in the layers. So back in 70s, the laying hens would lay about 200 eggs, and currently the same bird in the same period of time would be close to 350 eggs. And at the same time, we had the improvement of the feed conversion ratio from needing about 3.5 kilograms of feed 4 kilograms of eggs. We are down now to close to 2 kilograms. And there were at the same time also improvements in other traits related to sustainability, including livability. So what kind of traits are currently under improvement? Of course, we have egg production, which has different traits to it, like the number of eggs per hen, laying rate, and persistency. However, the challenge with the egg production is that it has some negative genetic correlation with both egg quality and some of the welfare traits. And also, it takes really long time to collect persistency data. So if you want egg production to 100 weeks, you have to maintain those birds for almost 2 years to collect that information. Next group of traits is feed efficiency. The problem here is phenotyping and how precisely you can measure the actual feed intake. Next group of traits is egg quality, which has both external egg quality like shell strength, egg weight, and shell color, and internal quality like yolk color or albumin quality. The difficulty with the egg quality is that you have to evaluate it over lifetime, because many of these traits decline or change in some way as the birds are aging. In the animal welfare group, we'll have livability, kill bone, and feather cover, which have the challenge of low heritability, and also the phenotyping may be difficult. Then we have robustness in terms of coefficient of variation of performance or reaction norms. The difficulty here is that really getting information on robustness needs some kind of challenge, so it requires multi-environment data. And then the last group of traits will be behavior, so we can have feather pecking, fear response. We also have positive behaviors like nest usage and perch usage. However, these are difficult to respond, to record on the scale, and to keep the data on the individual belts level, and they also tend to have lower heritabilities. Just to give you a quick overview what the modern egg quality could look like, it's based on the scanners, and then there are lots of machines involved, like the chromometer for shell color, then we have a scale for the egg weights, we have different measures of shell strength, and then we have different ways to measure internal egg quality. So it's quite a complex process to evaluate comprehensively egg quality. What kind of we're talking about? We are mostly in low to moderate range of heritabilities for different layer traits. For the egg quality, like shell breaking strength and egg weight, they can get up to higher upper end of the moderate heritability, and also some traits of related to feed intake. But as I mentioned before, the traits related to the livability and welfare, they will be on the lower side. Oftentimes less than 10% of the variation would be coming from genetics. So our second group of traits are the traits related to the animal welfare. The most important one, I believe, would be the livability. But the observed scale, when we look at overall all causes, has very low heritability. You may get a little bit better estimate if you use threshold type of models, going up to maybe 18% of the liability scale. However, if you look at mortality related to specific causes, you can get both better heritability and actually observable response to selection. And here I like to use the selection against Marek's disease mortality. And we have here one line that was under strong selection against these diseases. And we have survival on the y-axis and the time in the experiment on the x-axis. And when we look at the older generation in red, by the end of the experiment, less than 60% of the birds were alive. When we look 2 generations later, by the end of the same period, almost 90% of the animals were alive. And this was possible by both strong focus of the selection on this trait, and also in this particular line, there was a major gene that was segregating, which allowed this really significant improvement in livability. Another thing that is in the welfare group of traits is the indirect genetic effect. So there was a really important study by Bill Muir, which showed that if you select birds in individual cages, you may get undesired response with them getting more aggressive and causing mortality when they're put together in the groups. However, if you, instead of doing individual selection, put the birds in the family-based groups and just look at the more productive and higher livability families, you can simultaneously improve both egg production and livability. The theoretical framework for this model was developed by Peter Baima, and it changes the basic model of the phenotype from saying that the phenotype of the animal depends on its genetics and environment. It considers that in the environment there may be a genetic part. So the total breeding value will be direct breeding value of that animal and -1 times the indirect effect of the other animals that are in the same group. And what that model allows us to do is going from the 3 to 10% heritability with the standard model to estimating the total heritable variation for the survival with going up to 30 to 60% of explaining phenotypic variance. And there were also some selection experiments that were showing that this kind of selection using total breeding value can significantly improve livability over the span of 5 to 10 generations. And also that there are different genomic regions that are responsible for direct and the social effects. However, the challenge with this is that it requires quite specific family group structures in the nucleus breeding, and the birds have to be housed by family. And those groups can't be too large because as the group size becomes bigger, the model can have convergence issues. One trait in this group that I want to talk about would be feather pecking. And it has about 5 to 15% heritability. If we include this group-based variance, it can increase up to 40–50% of phenotypic variance explained. So the social effects account for 70 to 90% of the total heritable variation. So when a chicken is using feathers, it's not as much her own genetics that is responsible for her feather cover, but it is mostly the genetics or the aggression of the other birds that are within the same groups. And there were also some genomic regions identified to be associated with feather pecking. And those candidate genes were related to the dopamine and serotonin pathways, which kind of makes sense that the brain would be involved in the levels of aggression. So how can we phenotype this trait? The gold standard would be to have somebody sit and observe directly the birds. However, it's very labor intensive and may not be very accurate. Another way would be to look at the outcome, so score the feather coverage of the birds. And also you can look at the damage of other body parts, not just feathers, like toes, or score separately back versus vent. And newer approaches would include image or video analysis algorithms. How can we select for these traits? It would be probably multi-trait index and using group selection to capture social genetic effects. Also important here is to use for selection information from birds that are not beak trimmed, because that way they would express to the maximum extent their tendency to aggression and feather pecking. And you can't, of course, measure it in the single bird cages because there is no interaction between birds. However, you can measure these traits in either furnished multi-bird cages or in aviaries. So this is an example of what the feather cover assessment would look using pictures and cameras. So a bird with good feather cover would have nice uniform body temperature and be well protected from the external factors. As they start losing feathers, you can see that this bird is missing lots of the feather cover at the back, and there is lots of heat escaping from this hen in their back part of the body. So her feather score would be poor. And using the cameras in real life allows monitoring and automatic scoring of the birds and identification of the problems without having employees sitting and continuously watching the flock. Another trait in the welfare group would be beak length, and it has up to a moderate heritability. and it was shown that it is somewhat correlated with mortality and negatively correlated with the plumage condition. So the way how much longer this upper big part is from the lower one is going to affect how much injury the hen is going to inflict when pecking her penmates. Another group of welfare traits would be kill bone, and they can be scored on a scale from totally perfect kill bone with no damage, deviations, or breaks, up to really poor quality with multiple breaks, and this one is almost not the proper shape. So this can be either evaluated with the X-rays, or a simpler way would be to use palpation score, because these kind of fractures are going to create calluses, especially if then they are on the tip. It's possible to palpate them with your fingers. The heritability of the palpation score is pretty decent, showing that it's a trait that captures some of the genetic variation. And importantly, it's not very correlated with egg quality and egg production traits, with a little bit of exception of negative correlation to the early egg numbers. So the birds with very early sexual maturity tend to have more kill bone problems, possibly because their bone structure is not properly developed at the beginning of the onset of lay if they start laying too early. Another group of traits here is adaptive capacity. So you can measure fear response in birds by tonic immobility. So you take a chicken, you flip it, and then it's going to freeze. And you can measure how much time it takes for the chicken to start back moving and get active again. So that tells you how stressed or fearful they become and how quickly they are able to recover. Another stress-related or exploratory behavior would be open field tests. So you put a chicken in the unknown environment, in the new place, and measure how much time it takes for them to start exploring. And both of these traits have some level of heritability. Or you can also measure directly corticosterone levels in response to stress. It was shown that the selection for low tonic immobility duration, so quick recovery from stress, was correlated to reduction in mortality from feather pecking in the cage-free systems. Another thing that we have to consider in the breeding of the chickens is that there is significant genotype by environment interaction between different production systems. So the egg production in the cage is not 100% the same as egg production in the aviary. So what we have to do to breed chickens adapted for the target housing system is to use that specific system, or explicitly challenge them, or put the progeny testing or SIP testing in multiple environments. That G by E level differs between the traits. As I mentioned, egg number is one of the more affected ones. So egg number, egg mass, and production efficiency are going to be more affected by G by E, but traits such as body weight and egg weight tend to be more consistent and show lower extent of G by E. And now we're off to the last group of traits related to environmental efficiency. This chart shows that the biggest impact of layer system on the environment is related to the feed. And FCR or residual feed intake are both heritability traits, moderate heritability. and it is possible to get a response to selection, as I showed in one of the earlier slides. And FCR directly reduces the feed use per the amount of product, which also is related to the less land use and less production excretion of the nutrients. So feed production contributes approximately 85 to 90% of the greenhouse gases per kilograms of eggs, but still the management of manure and the farm energy are important for the remaining part of the greenhouse gas production. Cage system has the lowest production per kilogram of eggs, and the organic system is the worst for the greenhouse gases emission, primarily due to the lower feed conversion ratio efficiency in the organic system. Genetic improvement in FCR can contribute to a modest but real greenhouse gas reduction, with approximately 0.25% of production per year from FCR genetics in layers. However, the larger gains require combined genetic and management interventions. The traits that affect what environment would be also nitrogen excretion and phosphorus utilization. I'm not aware of any studies that would measure those traits directly in the layers. However, there are some estimates of heritability from broilers, and both of these traits were documented to have some level of heritable variation. A part that may be contributing to them is the host effect on the microbiome, because there is some heritability to specific OTUs that influence nitrogen fermentation and excretion profile, and the proportion of those OTUs in the microbiome can be heritable. So in short, the selection of RFI reduces feed intake for the amount of product, and that also leads to the proportional reduction in the nitrogen and phosphorus intake, leading to the lower excretion per bird. However, current limitation is that those excretion phenotypes require individual housing and total collection of all the excretions, and optimally, if you also want to include the CO2, that would require the total chambers and would be really costly to do this kind of experiments and selection on the large scale. And to have effective selection, we really have to go in the scale of at least thousands of birds, if not tens of thousands. Another trait that is related to the environmental impact would be persistency of lay. And that's because if we go from 72-week cycle to 100-week cycles, the rearing cost, which is the non-productive from the producer's perspective part of the lifetime, is amortized over 30 more eggs. So that gives us environmental benefit of about 10% reduction in the CO2 per dozen of the eggs by going and keeping the hens from 72 to 100 weeks. The challenges here are the physiological and immune system aging, and also shell quality declining with the age of birds can be a limiting factor. The way these challenges are addressed is by longitudinal phenotyping, so measuring both egg production and egg quality at different time points in life, at the early age peak and up to 100 weeks of production. And the last group of the 3 pillars is economic resilience. So while it is really important, the environmental impact and the welfare, if the birds, the production is not sustainable from the economic side, the producers are not going to work with that kind of birds. One of the problems here is the losing eggs due to shell problems. So you can lose, especially at the older age, up to 10% of eggs being downgraded due to shell quality. The heritability of shell strength is on a better side from the layer side. The problem is that there is some antagonism with the egg number and also egg weight. And however, the improving late shell quality with specific selection at 80 to 100 weeks of lay can improve the late cycle shell without affecting negatively egg production in the early part. And one of the traits in the quality that is relevant depending on the area of the world is the cuticle quality. That is a layer that is on the top of the shell, which impacts microbial penetration. However, in some countries, like the US, that shell is washed on the eggs, so we don't get the benefit of the cuticle. But having the cuticle level can allow storage of the eggs without refrigeration for a more extended period of time. Another group here would be genetic robustness, and it can be measured in different ways. One of the ways to assess robustness would be looking at reaction norm models. So we put animals in different and known level of challenge, and we look at the performance as a function of overall level of genetics of that animal, but then we can also estimate the genetic change with the environmental change. So we get average performance, and then we get estimates of the sensitivity to environment. Second way to measure robustness would be to look at the heterogeneity of variance. So we can look for the birds that have lower coefficient of variation across life or environments or challenges. And the last way is to do sire tests, progeny testing, or SIP testing in multiple, preferably more than 3 environments. And those environments can have different ways of challenges, such as heat, disease, or a poor diet. And that will allow either genetics that is regionally adapted to the challenges that are present in a different region, or we can look for the genetics of the birds that are robust against different kinds of challenges. Here is an example of what such challenge may look like. So this is an egg production curve, which normally the means would stay here and be in a level and then start declining as the birds age. However, here in period 12, the mean is a lot lower than it should be. It should be around here because there was a challenge at this point. And we can also see that not only mean declined, but also the variation increased a lot. So we can look at the birds. Are there birds that maintained the production for this period, or were they all equally affected? And we can calculate breeding values for this resilience trait. And here we have progeny of the birds that had high variance, so low resilience, and we can see that their production was really high, but then during challenge or around the challenge period went down to almost 0. And then many of them recovered thereafter, so we can see that the mean went back after the challenge. However, there were also birds that had low variance, but you have to be really careful to look at this trait without the context. So we have the birds that had high production and seem to be unaffected by the challenge. They continue to be high production, so they are highly resilient by the metrics of having low variance. However, there was this bird that had no low production no matter what. So this is not the kind of resilient birds that we want to have. If we look at diseases resistance, there are estimates of heritability for multiple diseases. The most successful selection, I believe, was done for the Marek's disease. But there is also shown to be heritable variation in other important poultry diseases. However, the challenge here is the access and the cost of data collection, because to do effective selection against diseases, having a regular challenge in defined conditions would provide the most accurate information. For some of the other traits, what we can do is just to obtain the data from the field and looking from the natural outbreaks, what's the genetic difference between the survivors and the birds that died from the infection. So what are the strategies for data collection for robustness? We have the birds in the optimal condition, thermoneutral in the nucleus, where we have the birds with the full pedigree. And for them, we can record all the production traits, welfare and quality traits. And these conditions allow them to express their full genetic potential for the production. We can send birds to a heat stress challenge and do that in the field test, and in that conditions collect also production and quality and mortality data. So these conditions are less controlled and they can be high variation. We can do natural or regulated disease challenges, and then either just simply look at the survival or clinical signs, or also look at the more detailed traits such as viral load or serological response. However, they tend to be pathogen specific, and in real life, birds can be affected by multiple pathogens, and there are also pathogens specific to the world regions. Another challenge that can be done would be low quality diet, and that can be done in the field test or also possible to some extent in the clean nucleus herds. And you can look at the feed conversion on the lower quality diet and also how the diet affects production and the egg quality. And you can also look and collect traits at the commercial multi-bird cages or aviary types of farms, where you can get the livability, feather coverage, and also get the egg number and egg quality. So my favorite part is genomic selection, because that's something that can help us, especially working with those low quality traits that are hard to collect. The concept of genomic selection became more popular with the Theo Vicens paper from 2001 and became more realistic for chickens when the genome was sequenced in 2004 and was actually implemented in both layer and broiler companies around 2010. There are available SNP chips with 50 to 600,000 SNPs that are used in the commercial breeding programs. And there are two main approaches. One is GBLAB based, where the pedigree based relationship matrix is simply replaced by genomic relationship matrix, and you can use the same models and get better accuracy. And currently, probably the most popular method would be single-step GBLAB, which integrates all the genotypes, phenotypes, and the pedigree simultaneously, even for the birds that don't have their own genotypes. Most of the gains over the pedigree blab are in the range of 20 to 50% increase of the accuracy. It also allows reducing generation interval, because you can select young animals that don't have data yet, which is important especially for those late cycle, so like the 100-week egg quality and the production to 100 weeks. The challenge here is a genotyping cost, and you also have to collect blood or tissue samples to extract the DNA and get the genotypes on large scale. And I'm sure you are all aware of the breeder's equation. So the genetic response is proportional to the intensity of selection, accuracy, and genetic variance, and inversely related to the generation interval. And genomic selection can help with all these aspects. So for the intensity part, you can make two-stage selection, so hatch more birds and then select them prior to housing. And you can also hatch more male selection candidates than we used to with the pedigree. Genotypes provide additional source of information, and that's especially important for the difficult and sex-limited traits, which for the layers are all the traits related to egg production and egg quality. With genomics, we are able to perform within family selections. So before genomics, the pedigree breeding values for the males from the same family would all be the same, because they would be based on the phenotypes of mostly their sisters and dams. So all those males would have the same sisters and dams. We would not be able to differentiate between them, so we would have to select the whole family. However, with genomics, we can get individual breeding values within families, even for the males, which allows us to do better inbreeding control by not selecting whole families. And it also allows, yep.
May I kindly inform you that we are already going late with your presentation.
Okay, so we go to the end.
Yeah,
so
thank you.
These are the take-home messages. So we need to make a progress with both welfare, environment, and economic pillars, and do it simultaneously. And we can use emerging and novel methods from both genomics and phenotyping to achieve that goal. Thank you.
Thank you so much, Anna. A nice presentation. Do we have time, Dr. Akram, for questions?
Yeah, we have 5 minutes for a Q&A
questions,
session.
okay. Yeah. Is there any question for Dr. Waltz? If not, I have a question for you, Anna. You elegantly mentioned that when you have the selection in group compared to cage, you have better production and better livability, right? Is that
not necessarily better? You can get information on the other aspects. So you get more accurate phenotypes in individual cages, but if you want to capture the social interactions, you have to use both environment. You have to provide the social environment.
You answer my question. I was thinking about the social aspect and that maternity trait, but you answer my question. Okay.
Yeah, so currently we use like all three types. So there are single bird cages, there are multiple bird cages, and there are aviaries. And you want to collect data in all those systems because that's what's relevant to the where the birds have to perform.
Very nice. Very nice. Any question for Dr. Walk?
Anybody have a question from Dr. Anna's presentation? Okay, thank you very much, Dr. Anna, for an excellent and insightful presentation. Your work particularly highlights the remarkable role of genetics and breeding in shaping the future of poultry production. I'm particularly very much impressed and very much happy to connect with you as I'm working in the poultry industry, in the layer industry of Pakistan. And we have Highland W80 strains, 1,20,000 birds we have currently rearing, and it's in the production period now. So thank you very much, Dr. Anna, once again. So, Professor Sami, we have now oral presentation session. We have two presentations in this session from China. So
Okay.
our first graduate presenter is Ali Hassan Nawaz, who is doing a PhD in Nanjing Agriculture University, China. His topic will be integrating transcriptomic meta-analysis reveals tissue-specific molecular strategies for heat stress adaptation in chicken. So it's over to you, Ali.
There's I
Yeah.
think there's a chat from someone, and also I would like to ask you, Dr. Akram, is 25 minutes the presentation of the student, or what is the time frame?
Getting very length. Yeah, we have 10 to 15 minutes for graduate presenters to present their presentation. So, Ali, you have 10 to 15 minutes for your presentation.
Okay, okay. Can you hear me? Is it
Yeah, we can hear you.
okay? Okay. Hello, everyone, and thank you for connecting from different part of the world. So here I am joining from Nanjing, China. So today I will share my work, my PhD work with you. So let me share my presentation. So I hope you can see the presentation
Yeah,
now. My topic is
perfect.
so the topic of my talk today is investigating the genetic and regulatory mechanisms of heat adaptation in chicken through multiomics. So here I divided this presentation into these 5 parts: background, objectives, methods, results, and conclusions. So first, let's move to the bit background of this research, why this is important, as all of us, we know that nowadays the world is facing the issue of uh, heat stress. So heat stress is like a global issue now, and we know that it's affecting the poultry production, as Professor Sami Dridi also working on heat stress, and he mentioned that how heat stress is affecting the chicken production, and also it like induces mortality in various regions. So here you can see that in different regions of the world. So it's estimated that in 2090, more than 25% of poultry flock will face dangerous heat. So these are the basically conservative, very conservative estimates. But the real situation is even worse than that. So here you can see the global average temperature from 1860 to 2024. You can see how in these coming years, now the heat stress is increasing exponentially. So that's why this is an issue, and how it impacts the poultry production. Basically, heat stress impacts almost all the organs, including liver, like liver fibrosis, and it also impacts the digestive system, immunity. It activates the heat shock response. So this is we already know that how heat stress impacts the poultry system, like its physiology, and at the end it affects the production. So in this study, the major threat is the global climate crisis, and it's impacting poultry production, and its impact is basically on reproductive performance, production, and immunity. And the solution, one of the solution, is to understand the genetic mechanisms behind the heat tolerance, because we know there are some breeds they are already heat tolerant, like indigenous breeds, most famously, the Fayoumi breed from Egypt is very known for the heat adaptation. So to address this question, we have this research design. We did genome-wide selection scans, multi-omics integration, and network modeling to get to know that genetic mechanism behind this. So in this study, we selected, we collected the blood samples, whole genome sequencing, we did population structure analysis, then we did selective sweep, functional enrichment analysis. You can see. After that, we moved to RNA-seq, and after RNA-seq, we also did EQTL mapping. So the study populations here, you can see in the map that we selected 2 chicken breeds from the hottest area of Pakistan. Mostly in this area, the temperature is 48 and also up to 50 degrees centigrade during some days in summer. And then we selected 2 breeds from Thailand, as we know that the overall weather in Thailand is humid and hot. So in Pakistan, it's hot and dry and humid. And then we also selected some breeds from China, South China, the humid regions. And then some for the control group, we used cold chicken breeds from the northern China. And red jungle fowl included as a reference group in this analysis. So after whole genome sequencing, we collected this data and we performed various quality control analysis. And at the end, we have 16 million high quality SNPs. So I will quickly go through the results because I have limited time. So basically, I divided results into 3 parts: whole genome sequencing of indigenous chicken breeds, then integrative genomics, selective scans, and transcriptomics. So here is we did classified the chicken breeds based on climate. You can see here in the table these chicken breeds from Pakistan. We divided into hot, dry from the Pakistan, and the area from the Xinjiang, China. We divided those breeds as a hot, dry. And then chicken breeds from Thailand and the South China, we divided them as classified them as hot, humid. And from the northern China, we classified those breeds as cold. So after that, we performed population structure analysis. And here you can see that in this PC analysis, these breeds, they make clear separation. Those you can see here, the red, in red color, they are hot and dry region chicken breeds. And then yellow chicken breeds, they are like hot and humid, and blue, they are cold area chicken breeds. So here you can see they are clearly making a visible structure. And then we also did the FST analysis to check the genetic differentiation among each breed, and a mixture analysis. And then we also did linkage disequilibrium to check the indigenous breeds. And here you can see that that jungle fowl is with the shortest LD. So this was expected. So basically, we did all this analysis to be sure that our selective sweep analysis will be more reliable. We performed genetic diversity analysis and in each chicken breeds, and they are same like the genetic diversity parameters and inbreeding. So here, the summary of this part of results that we characterize the population genomic architecture of 14 indigenous breeds, and basically we found that now our chicken breeds, we classified them in the correct way, and now we can move forward with our selection sweep analysis. And in selection sweep analysis, we did first analysis hot versus RGF because we want to be sure that we don't confuse the domestication signals with the heat adopted signals. So first we performed this selective sweep analysis between these hot population and RGF population, and we found some classical domestication genes like TSHR, BDNF. So these results show that we are on the right track. So then we performed the next group analysis like hot versus cold, and here we found some consistent genomic regions through different methods. We Like we applied 4 different selective sleep methods here, including FST, pi ratio, Thajima-D ratio, because we want to be sure that the signals we are finding, they are actually the real signals and they are conserved across different methods. So next we did hot, dry versus hot, humid. So here we also want to make sure that they are like, because hot, humid and hot, dry, they are quite different kind of like climate conditions. So we performed this analysis too. And after doing all this, so we only choose the conserved genes that we identified through all different selective methods, and we did functional enrichment analysis. Here you can see in hot versus RDF, hot versus cold, we found like quite interesting result that in all these genes, the 5 pathways were common or conserved across all 3 groups. Is adrenergic, GNRH signaling, apelin signaling, calcium signaling, and vascular smooth muscle contraction. So further, we focused on these 5 pathways, and using chicken GTEx data from chicken GTEx, we checked the base gene expression of these 25 genes. Those were enriched in those 5 pathways, and we found that these genes, they are like their gene expression was quite interesting, and they were correlated with each other, and they were mainly expressed in hypothalamus, heart, liver, and brain. So afterward, we saw the we did like correlation analysis, and we found that these genes, these 25 genes, their expression is correlated with each other. And among these, the 4 genes stood out: the ATP1A1, PLCB4, AKT3, and RYR2. So now we focused on these 4 genes, and we did these locus zoom only for these 4 regions, and you can see they are quite differentiated among different populations. After that, we also checked the allele frequency of only these 4 regions. As you can see, they have like the PLCB4 has 812 outlier SNPs and so on. Then using EQTL data from chicken GTEx for the same, from the same genomic regions. So we also found that in those these genomic regions, they have EQTLs mainly in liver, trachea, and immune tissues. And after that, we also did TWAS association analysis, and we found these genes, they are mostly related to cardiovascular traits. So this is also interesting because most of these genes, they were also enriched in cardiovascular pathways, and now the TWAS association also showed us the same results. Then we also did some FIVAS analysis validation to be sure, and after all that, then we did LFM analysis. So this is basically like a GWAS analysis. So to be sure about these genes and to find some exact SNPs, we performed this LFM analysis. And we found that these genes, they have some real SNPs in promoter region that might be helpful in heat adaptation. And after that, we also do the transcription factor disruption, and we found that heat shock factor 1, these genes like PLCB4 was related to the heat shock factor 1. So these are the interesting results. So then we now we have something like the PLCB4 and ATP1A1. They are mainly they have some kind of role in the heat adaptation. And after that, so the summary of the results that we found 25 high confidence genes and how they interact with each other, like GNRH signaling pathway, calcium signaling, and adrenergic, basically this neuroendocrine and cardiovascular nexus of pathways, and how they help to help the chicken to adapt heat. After that, now we have whole genome analysis, and after that, so we want to be sure about that. We want to get some picture about the transcriptomic also. So for this analysis, we use the RNA-seq data from different 11 different experiments of the heat stress, and then we did in liver, breast muscle, and in the heart. So in liver, we found these pathways including cardiac muscle contraction, insulin signaling pathway mainly enriched, and then in breast muscle, mostly the FOXO signaling pathway was consistently enriched, and same in the heart. And heart, we found the same pathways like apelin signaling and calcium signaling pathway, adrenergic. These are the same pathway we found in our selective sweep whole genome analysis. So then we found that after that, we also like did WGCNA to be sure about like we want to another line of evidence. So we found almost the same pathways. You can see apelin signaling pathway, adrenergic signaling pathway consistently enriched. So it means that there is something going on with these pathways when it comes to the heat adaptation. So after that, we only used those like highly conserved genes among DEG, like DEGs, and also the WGCNA hub genes. And then we did the same like functional enrichment analysis, and then we performed, we used the decouple R to get the picture like protein transcription factor activity or protein activity picture. And we found that most of these genes, they are related to like involved in MEF2A and heat shock factor 2. Mostly these two family of transcription, they are mostly responsible for the stress response. So from these, then we knew that there is something going on with these genes and these transcription factors, and further we validated these genes using four different methods, and we found that these transcription factors that consistently associated with these genes we found in these meta-analysis. And we did
Dr.
same
Ali,
for
excuse me, Dr. Ali, just to remind you, you have 2 more minutes to conclude your presentation, please.
Okay. So I will just skim through these results. We did same for the breast muscle, and we got like FOXO family of transcription factors. So as we did with the liver, we did same with the breast muscle, and then same with the heart. So you can see in heart, we also saw the activation of heat shock factor 2. And so this is the summary of these results. So basically this transcriptomics analysis showed us that mainly liver was responsible for the metabolic suppression, because during the heat, if the metabolism is more active, there will be more heat. So it basically, the liver activates insulin and PPAR axis to shut down the gluconeogenesis and lipogenesis to reduce the thermogenic cost. And the breast muscle, it mostly like to enforce cell cycle arrest and chaperone-mediated proteostasis, similar like liver. So it will the breast muscle will prioritize the sustaining. They will use more energy for the sustenance, but not for the proliferation. So this is the basic like concept, and the heart is deploys enhance calcium cycling, vasodilation, sustaining cardiovascular output for systemic heat dissipation. So then we can see that how these three organs like liver, breast muscle, and heart, how they are acting together and forming a nexus when it comes to like heat adaptation. So the overall conclusion from you can see here from the top here, like we found some genes, and we found some SNPs in those areas, like from whole genome sequencing analysis, that these genes, they were mostly related to MEF family and the heat shock factor family. And then we came to the transcriptomic analysis, that how transcriptome reacts to acute stress. And then we found that the reproductive resilience, cellular iron homeostasis, and cardiac output and vasodilation was the main, like in indigenous breeds, these are the main pathways or functions through which the indigenous chickens adopt heat. So climate selection reshapes PLCB4, ATP1 promoter regions and regulatory architecture, and calcium signaling and cardiovascular regulation. And these are some published articles in Poultry Science Animal from this study. Like our group is working on heat stress from many years. So this is These are the studies we published recently. And here at the end, I want to thank Professor Fang and my research team for their support during my stay and work in China, and also those who funded this study. And thank you so much for listening.
Thank you very much, Ali. Thank you for your nice presentation. I think we have time for a few questions.
Yeah,
Any question from the audience, please?
there is no questions from the audience side right now.
I have two
Yeah.
questions, Ali. My understanding that you collect tissue from different breed, from different region, from Pakistan, from China, and from Thailand, right? And you show some differences. You used RNA-seq and omics. And my question: how did you control or you distinguish between the effect of the breed themselves and the other factor? Like my guess, you didn't collect them at the same time. Like you showed that the BDNF, there's some difference. BDNF has a circadian expression within 24 hours. Then how you control that? The diet, the environmental condition, the time, how you distinguish between those effect and the effect of the breed?
Yes, Professor. We we know that there is also the ancestry and those things. There is we cannot be sure that these are only the environment signals, but we performed. There is an analysis like LFMM is same like GWAS. It reduces some. It has K factors. So we used K factors 11, like in admixture, we use K is equal to 7 to remove the ancestry effects. And then we also perform the LFMM to make sure that these are environmental signals. But still, we cannot be sure, 100% sure that these are environmental signals. So, and for
Yeah, go ahead, please finish.
And for the domestication signals, we excluded domestication, like you're saying BDNF and TSHR. We didn't include them because these genes, they are already known as very well-known domestication signals. So we excluded these genes from the heat adaptation.
My second question, you find a very nice, data related to lipogenesis. You mentioned that I think PCK1 to shut down lipogenesis, right, under heat stress condition. But there's some paper from Pierre-André, I think from Adisio and INRA. This is in the 1990s. They show that heat stress increase fat deposition in chicken because it enhanced lipogenesis. But you show the difference. Any comments on that?
Yes, Professor. Yeah, I know that this when I was writing the discussion, I came through this. But I think is because heat increases the fat deposition in the abdominal region. Because in one experiment, I also did I gave heat stress to chicken, and I found that heat stress actually increased fat deposition in the abdominal regions. But it's like intramuscular fat, it decreases. It decreased overall the quality of meat. This is from me. I think it increases actually the abdominal fat deposition, but it reduces
but that fat deposition comes from the liver. 95% of lipogenesis de novo fatty acids comes from the liver. The liver makes the fat and send it to the adipose tissue, right?
Yes.
Then,
but here is because maybe I also think about this. Maybe because this is in indigenous chicken is different. Because most of these, so that can be a reason. Yeah, but it's but we don't know about this. What's the truth?
Thank you so much. Very nice presentation. Very nice. I like it.
I have read your work a lot because I am also doing on heat stress and those. And now I will also read about that today. You mentioned about the water efficiency. I think it's very interesting, and I will try to also. So thank you, Professor, for your time. And
it's my pleasure. Thank you for your presentation.
Thank you very much, Dr. Ali, for such a wonderful presentation. We have one more question from Sadaf. Let me unmute you. Yeah, Sadaf. You can ask your question.
And you have
Sadaf,
one
can
from
you hear
Javad.
us?
And there's one question from Miral Javad, I think.
Yeah. Yes, Miral, you can ask your question now by unmute
Hello,
your mic.
am I audible?
Yeah.
Yes,
Okay,
I
great.
can hear you.
First of all, Professor Sami and Ali Hassan, you guys were really spot on my own research as well on heat and heat stress and because I know in the hotter regions of the world, this is the main issue that we are facing, and no matter the places, especially like Pakistan, they're using rudimentary things like uh, soaking up sand or cooling it with cooling the pens with ice or giving cold water to the chicks, but it's not really giving the full potential of the breed to reach the whole weight gain it's supposed to because of the heat stress, right? So I wanted to ask if I combine the sprinkler system and I combine it with, let's say, Ross 306, a very conventional breed, and I use drinking water. What should I do with the drinking water part to make it even better for the pens to take up energy and give a better FCR and give a better weight gain?
Miral, are you asking me or asking Ali?
I'm asking both of you. I would like both of your insights because this is the real problem we're facing in Pakistan.
If I may, there's no magic bullet. There's no one solution
Yeah.
for the problem. You can combine several. You can use the sprinkler. You can play around the water quality, the pH and all. You can play with the diet, vitamins, antioxidant, phytogenics, things like that, probiotics. I don't think there's one thing will solve the problem
Mm-hmm. But
because I think it's a multi-genic and multi-complex issue.
mm-hmm. But what about like just cooling the temperature around the chicken? Because like Ali Hassan said, the temperature is almost very high for maybe 8 months in Pakistan. So even
How you will cool the temperature?
yes, because sprinkler is one option for the temperature. Putting ice is one option. Giving cooled water to the chicks is one option. But what other things would you suggest?
As I said, this is a combination of things. You mentioned
Mm-hmm.
ice. Where do you make the ice of? To make a lot of ice for houses like 20,000 to 50,000 chickens, that's a lot of energy. Then you will have another problem. Not only water scarcity, but you have energy crisis, and we are living in an energy crisis today. You guys, you know that,
Yes,
right?
exactly. This is why I was
We have
studying
we
this.
have the gas here in the US for $4. The gallon is then you will I think it depends where you are, but I think it's a
Mhm.
combination of many
A
many
little bit
things.
of everything you were
Yeah.
saying.
Yes,
Sorry that I
and
didn't
by
give you
making
exact answer.
and by making more ice, we are making our environment more hot also. Like we're emitting more carbon, and I think that is not a solution from my side. That can be a short-term solution, but in long term, this would be a disaster. So we have to go for like as a researcher, we need to like focus on long-term solutions. So for short term, we can do, I think there is a lot of technology. So this is basically a technology question. So from our side, I think because this kind of research uh, like cannot be used in short term, so we have to do and find some genomic reasons that, and maybe in future we have answer to these questions, but no, we don't have any answers.
Okay, thank you very much, Dr. Ali
Thank
Hassan
you.
Nawaz and Miral. Hope you got the answer. So now we move to our last presentation of this session on genetics and genomics. And the presenter is Anika Imtiaz from Yangzhou University, China. She will present on mitochondrial haplogroups influence mitochondrial structure and function, oxidative stress, autophagy, and lipid metabolism of chicken hepatocytes in response to energy stimulation. So, Anika, you have 10 to 15 minutes for your presentation. Please conclude as soon as you can. Thank you very much.
Okay. Thank you so much, Mr. Sam, for inviting me for the presentation. First, I will share my screen. Is it sharing to everyone?
Okay.
Is my screen visible to everyone?
Yeah, we can see your screen. Please present mode. Okay.
Okay. Thank you so much. Good evening, respected professors, teachers, and fellows. And I'm very delighted to welcome you all for my presentation all the way from Yangzhou, China. My name is Anika Imtiaz, and I'm a student from College of Animal Science and Technology in Yangzhou University. So today, the title of my presentation is Mitochondrial Haplogroups Influence Mitochondrial Structure and Function, Oxidative Stress, Autophagy, and lipid metabolism of chicken hepatocyte in response to energy stimulation. These are the contents under which my whole presentation is structured. So first of all, I will start out with the introduction part where I will discuss the background and research content and significance of mitochondrial haplogroups. Starting off, I would first like to introduce what the haplogroup is. Based on the research, mitochondria are the maternally inherited organelles that don't go through any genomic, any kind of genomic mutations, and they are passed on to the next generations. So the lineage from which the a same group of mitochondrial DNA belongs, so we classify them as the haplogroups. Since the mitochondrial DNA lack homologous recombination, sequence changes and accumulate, causing mitochondrial DNA from the same maternal lineages to cluster into a mitochondrial haplogroups. For this study, based on the previous study, we have 9 haplogroups from A to I based on D loop sequencing. For my study, I used 2 haplogroups, A and E. Moving on, I will discuss the research gap and significance of this study. So first of all, we discussed the liver metabolism and liver as the metabolic center for the glycogen storage, protein synthesis, and as the Dr. Ali and Dr. Simi already discussed, that liver as the central organ for energy metabolism. The next is the model organism. We use the yellow feathered broilers in China that we received from the Jiangsu Institute of Poultry Research. Third, the knowledge gap distinction in mitochondrial structure and function between the A and E haplogroups is currently unclear. So this study is one of the contribution to understand how this all works. The second part of my presentation is research objective, where I will be discussing my research aims and hypothesis behind this whole study. So to investigate how mitochondrial haplogroups influence the maternal effect-related phenotypes, we first compare the mitochondrial structures and functions among the both haplogroups. Secondly, we examine the oxidative stress levels and reactive oxygen species production linked to the mitochondria in the both haplogroups. Third, we assess the autophagy capacity difference through LC3 protein expression. And next, we evaluate the lipid metabolism and fat deposition. Lastly, we elucidate the mechanism by which mitochondrial haplogroups affect maternal effects and production performance. Third is the research design, the experimental model, and the analytical methods. In this study, we did two types of experiments, in vitro and in vivo. For in vivo, we divided 40 hens into two groups, 20 hens in each group, and we have two different type of feeding models, the fasting and refeeding. During the fasting model, we gave them 12 hours food deprivation, and for the refeeding, we give 12 hours of fast and 2 hours of feeding. So we check the difference of this feeding upon both the haplogroups. For the in vitro model, we culture the 15-day-old embryo from the primary hepatocytes, and we also give two treatments. The first one we give with the glucose 50 micromole per liter, and the second one was the oleic acid to create the same steatotic condition in the hepatocytes. So we did multiple molecular analysis, just like the Western blot and oil redo staining, HE staining, reactive oxygen species analysis, and protein expression, triglyceride content through both type of studies. Next, I would move to the most important part of my presentation, the comprehensive findings from the in vivo and in vitro experiments. Starting off with my first figure, you can see it is the comparison of haplogroup A and haplogroup E. It's a picture from, it's a ultra structure using transcription electron microscope. So you can see in the top image that haplogroup A has a higher band and the haplogroup E has the lower, which means the shorter mitochondrial circumference, and that reduces the mitochondria and reduced mitochondrial associated endoplasmic reticulum membrane strength and fewer endoplasmic reticulum contact points. So under this nutritional stimulation, E group mitochondria has smaller with reduced endoplasmic content points, suggesting weaker fusion and organelle interaction capacities. Next is the mitochondria ultrastructure in the fasting group. In the fasting stage, you can see in the E haplogroup, there is increased mitochondrial compound and compromised structural integrity. So during the fasting, E group shows increased mitochondrial proliferation but compromised quality. Next, I have there is a picture from mitochondrial function during the refeeding stage. We can clearly see that the membrane potential similar between the both groups. There is no significant difference. And despite the normal membrane potential and reactive oxygen species, the severe swelling in E group suggests the compromised mitochondrial membrane integrity. The y-axis shows the count, while the x-axis shows the integrity of the fluorescence. So we can see the peak in the E group is higher as compared to the, in the A group is higher as compared to the E group. Next is the mitochondrial function fasting. You can see that membrane potential is lower in E group and trend not significant, similarly like the previous slide. And reactive oxygen species is low in E group. In the E group, there is not significant reactive oxygen species, whereas there are significant, a lot comparatively more reactive oxygen species in the A group. So these combination of increased swelling with trend of reduced membrane potential suggests the impaired mitochondrial stability during the energy deprivation. This is a Western blot image from the protein expression of multiple genes like MNF2 for fusion protein, respiratory chain protein, and complex 1MT and D1, LC3 for the autophagy, and SOD2 as the antioxidant. So there is during the refeeding stage, you can see the expression of the blots in these groups. So we see the baseline protein expression is similar between the groups. However, the combined structure data function differences may emerge at the activity level rather than the expression level. But you can see during the fasting stage, the expression is more prominent and reduced MNF under the fasting stage and the compromised mitochondrial fusion capacity in E group during energy deprivation stage. Next is the oil redo staining and H&E staining images from refeeding, and we can see that there is a significant difference among the haplogroup A and haplogroup E. And I also have the fluorescence images from both the groups, and you can have a look that E group is more viscous and significantly higher fat content than the A group. So the E group hepatocyte accumulates more lipid under nutritional stimulation and correlation with compromised mitochondrial function. Next, we have in vitro results, the reactive oxygen species and membrane potential during, you can see during the oleic acid supplementation, it significantly triggers oxidative stress in the E group and hepatocyte indicating haplogroup dependent differentiation responses to fatty acid overload. This is the protein expression in vitro during the fasting stage, and we can clearly see that the E group, the LC3 in the E group is significant, whereas the reduced autophagy in MNF2 and CYTB, no significant difference was observed. Under energy stimulation, E group exhibits a weaker autophagy, lower LC3 and impaired mitochondrial fusion. This is the blot expression of triglyceride content in the in vitro, the embryonic cells. During the glucose treatment, we can see no significant difference in the triglyceride content, whereas for the oleic acid treatment, triglyceride content increased in the E group. So combined in vivo data, this reinforced that E group has enhanced lipid deposition capacity, and consistent finding in both the models in vivo and in vitro showing greater lipid deposition. Finally, I would like to conclude my whole work. In conclusion, our study demonstrated that the mitochondrial hepatocytes significantly influence mitochondrial structure, function, and lipid metabolism. Moreover, compared with the E group, the A group exhibits a stronger mitochondrial fusion, autophagy, and stress resistance. In contrast to E group, mitochondria are more susceptible to stress induced damages and show greater lipid accumulation under energy stimulations. Also, these findings provide valuable insight into maternal genetic effects and may support future breeding strategies, nutritional interventions, and prevent the metabolic diseases in poultry. So this was my research work, and I would like to thank my research group and the funding project under the National Science Foundation for supporting this work. And I'm open to the questions if you have any.
Thank you, Annika. Thank you so much for your nice presentation. Any question from the audience for Annika?
There is no question in the chat box right now.
I can ask. We need to ask her, right?
Yeah.
Yeah. First, out of curiosity, you showed hepatocyte and in vivo liver using the LC3 antibodies. And in vivo, you have only one band of LC32. In vitro, yeah. Sorry, Anik, my first question, you showed immunoblot for LC3 2. In vivo, you have only one band of LC32. In vitro, you have the two bands, LC31 and LC32. Do you have any idea why that difference? Why you didn't see two bands in the in vivo? You are unmuted. You are muted.
Nika, are you on the board?
Yes, yes. Sorry, sir. I have a connectivity issue. Can you please repeat your question?
Your LC3 immunoblot, the Western blot that you nicely showed, in vitro you have two bands, LC31 and the LC32. In vivo
Yes.
you have only one band, and you said that is LC32. Do you have any idea why that difference? Why you didn't see two bands in the in vivo tissue?
Because in the embryonic stage, we selected for the in vitro model, we selected the hepatocytes at the 15-day-old embryonic stage. So there may be some, the subunits of the protein. So maybe we see for the in vitro, we see two bands, and for in vivo, we see one band.
Okay. My second question, you said that refeeding increase the number of the mitochondria. Does that mean increase of the biogenesis? And what is more important, the number of the mitochondria or the size of the mitochondria?
So these two are different things. The number of mitochondria means that there is more energy synthesis in the cell, in the hepatocytes. And so I showed that there is increased number of mitochondria after refeeding, which means that when we give a fasting, when we give a 12-hour fasting and then 2 hours of feeding, the energy synthesis and energy regulation was more better than during the whole fasting stage. So I think the number of mitochondria, more number of mitochondria is more important in the cell, as it
Okay. And what
regulates the more as it regulates the more energy, and also the autophagy, the mitophagy of mitochondria contributes to the self-defense system. So more number of mitochondria in the cell leads to the more defensive response.
Okay. Can you tell me a little bit more about the mitochondrial swelling assay? What is this assay? How did you analyze the swelling of the mitochondria?
I'm sorry, I don't understand your question.
No, in your material and method, you mentioned mitochondrial swelling assay. What is that assay? Can you tell me a little bit more about it?
I'm sorry, Professor, I'm unable to answer this question at this point.
That's
Maybe
okay.
I will have.
Yeah, no problem. No problem. Thank you.
Okay, thank you
Dr.
so much
Akram is
for your
yours.
time and for the questioning. Thank you so much, and I feel great meeting you all here. And goodbye from my
Thank you.
side.
Thank you,
Thank
Nika, for your presentation. So now I would like to ask Professor Dr. Sami Dedi to please conclude this session with the very short remarks, and with the token of appreciation to our distinguished keynote speakers and to honor presenters of this session. Professor Sami Dedi, it's over to you, please.
Yeah, thank you so much, Dr. Akram, and thank you for the organizing committee for inviting me first, and inviting the outstanding keynote speakers and the outstanding PhD student. I think we have a really nice session that covering from genomics to omics, from layers to broiler to heat stress to mitochondrial organelle and all that. I think it is a successful session. We didn't have much questions from the audience, but please, if you have a question, I think you have the contact information of the speakers. Feel free to send email. This is, I think, is a good opportunity for all of us to networking and to share. Heat stress problem is not a problem of Pakistan or the US. It's a global problem, and no one person or one country can solve that problem. I think this is a good opportunity for future collaboration, working together, and to do that, we need to reach to each other and share idea. And I hope that we will have more collaboration in the future. And with that, I stop here, because I know that I think we took more than what we are supposed to have in terms of time, and probably there are some other session after us. Thank you all, and I really appreciate this opportunity. Thank you.
Thank you very much, Professor Dr. Sami Dedi. I'm pleased to ask you to give a certificate of appreciation to our keynote speakers. Professor Song unfortunately cannot join us today. So the first certificate of appreciation goes to Dr. Kate Sutton. The next certificate goes to Dr. Anna Wallet from the Iowa State University. Thank you very much, Dr. Anna. And the last but not the least, to Professor Dr. Sami Dridi, who we really express our deepest gratitude for your outstanding leadership to our GRC 2026. So once again, Professor Dr. Sami Dridi, thank you very much for joining us today.
Yeah, it is my pleasure. Thank you.
And the last awards goes to oral presenters. The first certificate of presentation goes to Dr. Ali Hassan Nawaz.
Very nice.
And the next goes to Anika Imtiaz. So with this, we now goes to the end of our day one. We have explored critical topics in poultry nutrition, alternative feed resources, poultry diseases, gut health management, genetics and genomics. The discussions highlighted the importance of integrating scientific discovery with practical solutions for sustainable poultry production. And I really would like to sincerely thank our keynote speakers, session chairs, oral presenters, collaborators, sponsors, and media partners, and participants, particularly for their contributions. Tomorrow, we continue with discussions on poultry productions, systems, welfare, artificial intelligence, and poultry products technologies. So we look forward to welcoming you again. And thank you, and have a wonderful evening in Pakistan, and have a wonderful day to Professor Sami and Dr.
Yeah, thank
Anna
you so
Wallet.
much. Thank you. Take care. Bye-bye, guys.